Alrighty, we might get started. Thank you very much for joining today, Hatch House's webinar. We've got some regulars in today, some new people too, which is wonderful. I just wanted to spend the first minute just introducing myself. My name is Alex Langfield. I'm the Director and Senior Vet at Hatch House. We've got Jess, our lovely admin coordinator, brilliant mind of all operations within Hatch House. And we've got Amira, our recently joined vet who is located in New Zealand. So today there's been a lot of discussions and a lot of webinars about gut health, but for me, I find them quite technical and sometimes a little bit over my head. And I can only imagine they can go over the heads of a lot of other people. So I wanted to sort of bring back some basics to what gut health means and give you a sort of a vet's perspective on gut health. Now, there is a Q&A session or the chat. So if you've got any questions, put them in there and Jess will be monitoring that as we go. Otherwise, put your hand up. Happy to answer questions as we go along. Just to set the scene, I just wanted to sort of let you know we'll be doing a bit of a dive into anatomy, into poultry gut health, examining not just the process within the digestive tract, but also the significance of the processes. Oh, there we go. Also the significance of these processes for performance, welfare, and the longevity of our long-lived birds. I'm not a nutritionist, so this is definitely by no means going to be groundbreaking on the intricacies of poultry nutrition. But what I'm going to try and do is share some insights from a vet's perspective and also from an operational perspective on how the gut is really integral to everything we do in our day-to-day. Now, before we go any further, I just want to make sure we've got our terminology aligned because I'm going to be talking in some of these terms interchangeably throughout this next hour. So the GIT, also known as the gastrointestinal tract, is the full digestive system from the beak to the cloaca, and the digester that I refer to is that feed material that moves through that tract. You'll then also hear me talk about GALT, or the gastrointestinal lymphoid tissue, and that's basically the immune component within the gut wall. We have the microbiota, which is the microorganisms themselves that live within the GIT, and we have the microflora, which is all organisms. The microbiome is essentially that entire ecosystem, the microbes, their genes, and how they interact with the bird's intestinal system and how that interacts with the overall bird health. And finally, the word dysbiosis. So symbiosis means balance. Dysbiosis means the loss of this balance. And so when this balance is lost, this is when those bad microbes start to outnumber the beneficial microbes. Now we've set that up. Pretty much the gut's performance depends on three interconnected factors. So first we've got the anatomy and the structure of that bird's digestive system. The shorter tract, the high efficiency makes it really unique compared to a lot of other animals that we have. The second component to this is the microbiota. These organisms, they're not just passengers. They influence the digestion, they influence the immunity, and even the behavior of our birds. And third, obviously, is the diet. The ingredients, the particle size, and the nutrient balance determines what reaches that lower gut, and then which microbes really thrive in that lower gut. So the gut is far more than just that digestion tube. It's a multifunctional organ. So it handles the digestion, both mechanically in the gizzard, but also chemically via the enzymes that it produces. It's responsible for the absorption of nutrients in our water. It's involved in the secretion or the releasing of enzymes and mucus, which lubricates and protects that lining of that intestine. And it plays a role in regulating and coordinating hormones, neural signals that really control that appetite and the digestion within our bird. But probably the most important is it is the center of immunity. It houses the GALT and it houses the goblet cells that defends birds from infection. And we'll get into a little bit more detail as we go further along. So one thing about bird's digestive tract is it is much shorter than mammals. feed moves really rapidly from the crop to the gizzard and then into the small intestine. So our crop here at the start is basically that storage unit. There's no chemical or mechanical digestion that occurs in the crop. It is, when you open the crop, it is as the bird has eaten it. It then moves into the gizzard, or the proventriculus, and then the gizzard. And the gizzard is essentially the powerhouse of this whole system. It provides both a mechanical grinding and also the start of some mechanical digestion via the enzymes that are produced through the proventriculus. Once that material then passes through there, we start heading into this small intestine, or the duodenal loop, through to the ileum and the jejunum, then through to the colon. And it's within this small intestine, with its villi and its microvilli, is where that majority of the nutrient absorption actually occurs. Now, a bird has two seekers, or seeker, and they play a role in fermenting any undigested fibre. It also supports a really diverse microbiota that contributes to the immune defense of the bird. So ultimately, efficient nutrient depends on a really healthy structure of all these systems, balancing the microbiota, minimizing inflammation, and to understand how that absorption happens, we're going to look closely at the importance of microvilli. So the villi, or what we refer to as like finger-like protections that are microscopic, and they line the intestine. Their role is to increase the surface area available for that nutrient absorption. So think about it here. We've got the microvilli, and the longer this microvilli is, the more these little enterocytes, or these cells, have contact with the digestion that's moving through the gut. Each villi is layered by this single epithelial cell, which absorbs our amino acids, our sugars, our fatty acids from that digestion. And then within that, we've got all these capillaries and lymph vessels that transports that nutrients from the digester into the rest of the body. In a healthy bird, the villi are tall, they're slender and uniform, they're covered really tightly with these epithelial cells, and that structure allows for maximum contact. But when the villi are damaged, whether this is like a coccidial infection, Clostridium perfringens, mycotoxins, feed irritation, that surface starts to collapse, which is what we've seen here. So not only are we losing the height of the villi, but the villi are also spreading out. So we're not getting that contact with the digester that we need. So if that's the case, the epithelial cells can die, or they can detach, and the villi then become shortened and blunt. And this crypts at the base, so this part here, deepen as the gut tries to replace those cells. Ultimately, the result is less surface area, slower nutrient uptake, high nutrient loss into the lower gut because you don't have the ability to absorb it, and it feeds that unwanted bacteria. So, like I said, the villi is lined by enterocytes, and that secretes those digestive enzymes and absorbs those nutrients. They also release hormones and antimicrobial peptides to regulate appetite and control pathogens. So its health, or this villi health, is critical to the overall health of our bird. So, oh no, I've lost a photo. Oh well. So the real success of digestion depends on how intact and functional that gut lining, or that villi, remains. And this is what we call gut integrity. So these cells are joined together by really tight junctions, and they're specialized protein structures that act as like welders between the cells, and they hold these cells really tight. Their job is to make sure the nutrients move through the cells while keeping bacteria and toxins from slipping between them. So basically, we've got these two cells locked really tightly together, and it's allowing what needs to pass through into the bloodstream through, but it's stopping the larger cells, or the larger bacteria and toxins, from also getting through. On top of that, the villi is also coated with like a protective mucus layer, and this again protects those pathogens from slipping into the bloodstream and into our lymphatic system. When everything is healthy, this system works quite perfect, and the gut acts almost like as a really well-sealed filter. Nutrients flow in, waste moves out, and essentially the immune system remains quite calm, and growth and production continues. But when the bird faces inflammation from whether that's infection or mycotoxins, nutritional stress, these tight junctions start to loosen up and they start to peel away from each other. And what this then causes is this mucus layer thins and that barrier begins to leak, what we refer to as leaky gut. Once that happens, the bacteria and toxins that normally were being captured by this mucus layer and by these tight junctions can now filter into the bloodstream or into the lymphatic system. And the bird's immune system immediately reacts. It diverts energy away from growth and egg production just to fight the inflammation that's happening due to these tight junctions being pulled away. So maintaining this gut integrity isn't just about avoiding an enteritis, it's ultimately about preserving the bird's entire nutrient economy and also the general health and the prevention of bacterial infection translocation. In field terms, what does this actually look like? Well, it looks like wet litter because we've got malabsorption. Remember those flattened villi? They're not getting absorbed. We've got undigested feed coming out the back end. We're getting a lot of fluid loss because this water isn't being uptaken by these cells. We get some uneven weights and our uniformity goes a bit haywire. And in layers, we get weaker shells or more seconds because that calcium uptake is not being produced. So all the work that you put into giving them a balanced diet, we're losing a lot through the back end. So you can sort of appreciate that the chicken's gut isn't just one uniform space. It's made up of really distinct regions, each with its own pH level, oxygen concentration, and its own microbial community. So starting at the front end, we've got the crop and the proventriculus. These are really acidic and oxygen rich. So the low pH, typically around that 2.5 to 5, suppresses those harmful bacteria really early on and begins that chemical breakdown of feed. Now, the dominant microbes here tend to be aerobic, meaning they love oxygen and they need oxygen to survive. Now, as the feed moves into that lower intestine, these oxygen levels now start to fall and the pH rises just slightly to around 5.5 to 8. And this region supports what we refer to as facultative anaerobes, meaning bacteria can live with or without oxygen. And some of these bacteria include lactobacillus or enterococcus. And these organisms are key players in the digestion and the immune signaling. And finally, once we get to that seeker or the bifold seeker, the conditions really change quite dramatically. Oxygen is completely absent by this stage, and the pH really stabilizes at about 5.5 to 6. And this is where our obligate anaerobes dominate. So this is where bacteria that can only survive in oxygen-free conditions, so no oxygen present. They ferment undigested fiber and protein, and they produce our short-chain fatty acids like acetate or propionate or butyrate. And these short-chain fatty acids do a couple of important things. They provide direct energy back to the intestinal cells that have been doing all the work. They lower the gut pH to hopefully inhibit some of our pathogens, and they really help strengthen that mucosal barrier. So the gradients in pH and oxygen they're not just background conditions, they really shape that microbial landscape. And when that environment shifts, so if feed pH rises due to a low acid content or maybe excess undigested protein reaches the seeker, the microbial balance also then shifts too. And we start to see a bit of that dysbiosis, the unbalanced microbiota within our gut. In practical terms, managing these gradients is really in just ensuring proper feed structure to regulate that passage rate, slow the passage down, allow the absorption to happen so we're not getting undigested protein reaching that seeker, creating issues with our microbiota. We need to support our gizzard acidification with fibre, organic acids, providing lower pH water to provide that acidification that it needs. Now, onto our second component, which is our gut microbiota. It's an ecosystem. So chickens host about 700 to 800 different bacterial species. And for every cell in that bird's body, there's roughly 10 bacterial cells living inside the gut. So that's a pretty powerful reminder that the gut isn't just a simple digestive tube. It's a living ecosystem. It's a forest of microbial, bacterial organisms. It's essentially a fermentation chamber filled with bacteria, fungi, viruses, protozoa, and they don't just occupy space. They're partners in that bird's biology. They break down nutrients. They produce key metabolites like our short-chain fatty acids. They provide what we call colonization resistance, meaning they crowd out or they suppress some of our harmful bacteria before they can cause disease. So our goal in poultry production is really to maintain a steady, balanced microbiota that supports nutrient utilization, prevents disease, and protects that gut wall from inflammation or infection to prevent that translocation or the leakage of these pathogens. But here's the key: the balance is largely driven by diet. It's diet's consistency, its quality, even small changes in raw material composition. And that could be small changes from diet to diet. So for a broiler, guys, our starter, grower, finisher, withdrawer, our 4 diets within a 50-day period, small changes within that can cause this composition to change. So talking about that colonization aspect of the gut, when a chick hatches, its intestinal tract is all but sterile. There's virtually no bacteria present. But within just a couple of hours, that can change quite dramatically. So as the chick begins to breathe, it begins to eat, it begins to drink, bacteria from the shell, from the hatchery, from the litter, from the feed can start colonizing this gut. And within the first few days, we mostly see fast-growing, oxygen-tolerant bacteria, things like Enterococcus, E. coli, Streptococcus, start becoming pioneer species. They thrive in that oxygen-rich environment of a gut that's essentially non-colonized. They consume the oxygen, they produce metabolites that make conditions much more suitable for the next wave of microbes. In a perfect situation, at 7 days of age, the gut matures, oxygen levels drop, and we start to see a transition where Lactobacillus species becomes more prominent. This really helps to acidify the gut, strengthens mucosal barrier. And by that 2 to 3 weeks, essentially that microbiota in the small intestine has completely stabilized. And the ceca, well, the bird's main fermentation chamber, has now developed that rich community of bacteria. Now, this early colonization phase, really within the first 12 to 24 hours, is absolutely critical because you want the right species to establish themselves. They shape that gut environment and how it transitions and adapts. So if the wrong bacteria dominate early, for example, through delayed feeding, maybe a temperature stress, early antibiotic exposure, the microbiota can shift towards a dysbiosis from the very beginning. And then that imbalance has now colonized and will persist for life. Oh, sorry, excuse me. Where are we? Okay. So the very first feed that chick receives acts like a signal to the digestive system. It switches on that gut motility, it stimulates that enzyme secretion, and it triggers the growth and an elongation of villi. There's plenty of studies out there to show early feeding of chicks will result in a longer gastrointestinal tract compared to chicks that have been withheld feed for 12 to 24 hours. Now, a longer gut means more villi, means more enterocytes, means more absorption overall. So it's going to be best for us. So the earliest you can get these chicks on feed and water, the better. Now, what we feed also matters, and that's pretty obvious, right? Diets with a balanced amino acid profile, moderate fiber, good starch, all support digestive efficiency and healthy microbial population. Overly fine or highly processed feeds can really speed that digestive passage up, which then reduces the mechanical stimulation. So our gizzard, remember, is our powerhouse of this system. If it's got nothing to grind, its stimulation won't be there. So we won't build gut tone. we will have this high passage of digestion moving through the gut. So it's sort of washing across the villi rather than staying and being sticky and allowing that absorption to occur. And then, so we're really trying to set up like a consistent, clean, well-balanced starter diet to really set up this digestive environment. Now, one of the most misunderstood nutrients, and again, I am not a nutritionist, this is just my take on it, fiber's been considered a filler in a lot of cases, something that diluted the energy in our diets, reduced our performance. But now we know that the right type of the right amount of fiber can be one of the most effective tools for improving our gut health and stability. So fiber does much more than adding bulk. It shapes the gut motility, it influences microbial diversity, and even can affect the bird behavior, particularly in our long-lived birds like our breeders or our layers. So let's break that down a little bit. Insoluble fiber. So this could be like from coarse grains, can stimulate that gizzard activity, develop that muscle within that gizzard and allow the gizzard to do the mechanical breakdown. A well-developed gizzard slows down that digestive flow immediately. It improves that muscle and the integrity of our intestinal tract, and it promotes a much more acidic environment. And that is beneficial because it suppresses our harmful bacteria. Now, moderately fermentable fiber, like canola meal, provides food for our beneficial microbes, particularly for lactobacillus. Now, as these microbes ferment fiber, they produce those short-chain fatty acids that I was talking to you about, mainly acetate, propionate, and butyrate. And these short-chain fatty acids fuel those intestinal cells by reducing, because they reduce inflammation and they reinforce those tight junctions between those cells. So it's essentially strengthening that gut barrier function. But as with most things in nutrition, balance is key. Too much soluble or poorly managed fiber, especially from things like barley or oats, without some enzyme support, can increase our viscosity, slowing that passage rate to a point where it can be detrimental and we're not getting, the satiety is there, we're not getting that intake that we need. And so that can set up a perfect condition for a dysbiosis or a wet litter scenario. So our high soluble diet, like our barley, our oats, increases that digestive viscosity. It makes the intestinal contents thicker, stickier, slow down passage rate, which can reduce that feed intake. In practical terms, you'll see this as quite wet, like wetter litter, maybe some inconsistent growth when that soluble fiber level gets too high. Where moderate levels from ingredients like whole wheat do the complete opposite. They reduce the viscosity, but keep the digestion moving, and they stimulate that gizzard. So that mechanical action improves that feed grinding, increases acidification, supports better enzyme activity further down the track. This table essentially summarizes some of the main fiber sources that we use in broilers, breeders, layers, and how they behave in that gut and what their real world effects are on the digestion and the performance. So starting at the top, we've got wheat, probably the most accessible insoluble fiber source we've got for both broilers and layers. High in energy, used as both a fiber source as well as mechanical stimulation of the gizzard to improve gut motility and helps reduce that wet litter. We typically in broilers will, can put it in between 5 to 10% solely to help reduce the digestion movement and allow that gizzard to become acidic and develop that muscle bound, that muscle development. We've got oats and oat hulls. These contain our soluble B-glucans. They really support beneficial fermentation in the seeker, but if the levels are too high, they can cause that digestion to be really sticky and that water doesn't get absorbed, so then we get wet litter. So we use them sparingly or at really low doses. Canola canola meal is a really interesting one. It contains a mix of both soluble and our insoluble non-starch polysaccharides. The fermentable portion supports the short-chain fatty acid production, particularly butyrate, which again fuels that intestinal cell, helps maintain our mucosal health. we've got lupins. They provide some insoluble fiber, some lignin, which adds some physical bulk and stimulates that motility. So that's fiber, but what I'm really interested in is in our protein component. So in simple terms, every gram of protein that enters our gut, enters the chicken's gut, is shared between two systems. It's shared between the bird's own digestive enzymes and also the microbial population that lives alongside them. So a balanced amino acid profile supports the building and repair of that intestinal lining, strengthens the immune system, maintains the tight junctions between epithelial cells, and also really supports that microbial population, because protein is one of the main energy sources of the microbial population. A high quality, highly digestible protein, properly balanced for amino acids, supports that strong villi and stabilizes that microvilli. When crude protein is too high, we get some issues. So when undigested protein escapes absorption, whether digestion's been too quick, either there's too much protein to absorb, or we've got some problems with our enterocytes and that leakage happening, it moves into that hindgut, the seeker, and that becomes a food source of pathogens down there. So bacteria like Clostridium perfringens, E. coli, they thrive on excess protein, leading to what we call a protein fermentation. So instead of producing our beneficial short-chain fatty acids, these bacteria generate toxic metabolites, ammonia, amines. These compounds irritate that intestinal lining. It disrupts that microbiome balance and triggers inflammation, and we end up in this circling effect. Because as the intestinal lining gets damaged, we get less protein being absorbed, the digestion moves, causing more inflammation, causing the microbiota to become more unsteady and unbalanced, and we end up in this really nasty sort of cycle of events. On farm, you'll see the results really quickly. You'll have wet litter, you'll have high ammonia within your sheds, you'll have uneven growth, you'll have feed conversion losses, because this energy now gets diverted to managing the inflammation within the gut rather than being put towards body growth. And we are at a much higher risk of necrotic enteritis or a dysbacteriosis because of this. So the next question is, well, how do we get the benefits of protein without the gut penalties, like without having issues within the gut? Well, the answer really relies in optimizing the utilization. And this isn't just reducing crude protein, but making every gram of protein in the diet work harder for what we need it to be. So the first step is to focus on the digestible amino acids, not the crude protein percentage. Birds don't need more total protein. They just need the right balance. That means paying attention to sort of our key amino acids, supporting with potentially synthetic amino acids can really dramatically improve that gut health while allowing a small reduction in the overall crude protein. Using high quality protein sources is probably one of the most critical parts. So if you're using soybean meal or canola meal, making sure that they're consistent and that they provide that strong amino acid profile that your nutritionists are programmed for. And then third, applying some enzymatic support. So proteases, some non-starch polysaccharide degrading enzymes to unlock some of that protein fragments that would otherwise escape digestion and be passed out the other end. Together we can really, together these tools can really ensure that more of the protein is absorbed rather than having to add more protein to get the outcomes that we need. Now, water, it's honestly probably the number one most forgotten nutrient that we've got, because it's really easy as a vet and as a nutritionist to focus on the protein and the energy and the additives that we can put in. Now, every bird drinks roughly twice as much water as they eat feed to their weight. And so water drives that digestion, the nutrient transport, and the body temperature regulation. So when water intake drops, feed intake almost immediately will drop as well. Dehydration reduces that enzyme activity, it slows digestion, and in layers can directly impact that egg production. The key point is birds really need clean, cool water available at all times, because without that, we could be creating ourselves a bit of an inflammation or dysbacteriosis. Now, the water system itself can become one of the biggest sources of bacterial contamination. Now, just think about what the birds will be drinking. Inside the pipes, bacteria can attach to the walls and form this sticky layer of biofilm. Now, if the birds are drinking this water and we get these occasional little bacteria sort of slothing off this biofilm, that's going to be entering that gizzard. Now, if we haven't already programmed our gizzard to be highly acidic and of decent mechanical strength, we're going to allow this pathogen to pass through the gut. Now, if your gizzard's not performing the way it needs to, it's likely that your intestine is also not doing it. So there is a chance that these bacteria that the birds are consuming are now going to pass through those tight junctions that are starting to weaken, and that mucosal layer that's starting to thin out. So while it won't necessarily directly impact on the gut quality, if we're not managing this, we're introducing higher challenges to the birds, and challenges that the birds may not be able to filter through. So the two things that are particularly important when you look at water quality is your coliforms. And coliforms, look, they're present in nature. They occur in all human and animal waste. And they're not necessarily considered harmful themselves, but their presence can indicate the presence of other harmful diseases because they almost act like carriers. E. coli is a big one. It's found only in the intestines of humans and animals. So most strains are harmless, but some can cause gastrointestinal illness for humans, but also for our birds. So the presence of E. coli is a strong indicator that we've had fecal contamination. Now, with fecal contamination comes nasty bacteria like E. coli or enterococcus or spotty liver or fowl cholera that is entering our bird and potentially has a chance of not being filtered through these tight junctions. So this slide shows a typical water analysis report from a poultry farm, measuring our microbiological quality of the water. So here we've got what we're trying to achieve is an E. coli of 0. We want no fecal contamination of water. And then secondly, we're trying to achieve a less than 50 coliform number, meaning that, there is a contamination, a bacterial contamination there, but we want it less than 50. And the total colony count, meaning the total bacteria grown on the plates, should be less than 1,000. Now we can see here that we've achieved no E. coli here, which is great. And we've achieved a really good heterotrophic plate count, or that total colony count. But what we haven't achieved is these coliforms. Here we've got E. coli. Now, can you imagine your birds drinking that? They are taking in 56 CFU per 100 ml that they drink. And they are drinking, sometimes upwards of 250 ml a day per bird. So they're going to be consuming 2.5 times this E. coli units per day. That's a lot for the bird to have to deal with. And then our minerals and our pH effect really plays into account here as well. So the lower water pH, slightly acidic, helps suppress that bacterial growth, not only just within the water, but also helps suppress the bacterial load within the gizzard, because you're already giving them a head start. They're not having to titrate down to a low pH. They're already starting at a low pH. So using things like an organic acid or acidifiers creates that environment to discourage pathogens. But on the other hand, minerals can actually feed bacteria. So E. coli, the one essential mineral that it cannot produce on its own is iron. So if you've got a high iron content within your mineral component of your water that you're feeding to your birds, you have highly likely you are growing E. coli, probably better than you're growing chickens. So even small increases in dissolved iron levels can stimulate E. coli growth, particularly inside these biofilms, which meaning that you're increasing that load that these birds are exposed to. So essentially manage water like you would a feed ingredient. So when you buy feed from, an approved feed supply, you're expecting it to be salmonella free. You're expecting high quality feed ingredients. You should be managing your water in the same state that you are your feed inputs. We tend to test and fine tune feed really constantly, but water, honestly, we tend to forget about. We might do it once every 6 months, and it has just as much influence on our gut performance as our feed, particularly when they're consuming over twice what they would be eating. And the reason why this is all important, and the bit that I get really excited about, is our GALT, our gut-associated lymphoid tissue. Did you know that 70% of the chicken's immune system sits within their gut wall? This network is called the GALT. It's called the gut-associated lymphoid tissue. You can think of it as a bird's internal system that is constantly sampling what's coming through that digestive tract and deciding how to respond. It includes specialized immune structures like Peyer's patches, and sometimes if you do some postmortems, you can actually see these Peyer's patches rupture during the euthanasia process. They are like little red dots all through the intestine. The cecal tonsils are located near the junction of the ileum and the ceca, and the diffuse lymphoid nodules scattered all along that intestinal wall. These tissues are packed with immune cells, our B cells, our T cells, our macrophages. Our macrophages are essentially the ones that recognize bacteria and go and eat them up, and our B cells and T cells are the ones that recognize them and send the alert out. When the system detects something really harmful, it mounts that rapid localized immune response. So it releases antibodies, it activates inflammation, and it produces cytokines to try and control that infection. And when it's functioning well, the GALT knows when not to react, when it's okay to leave be. It builds tolerance to harmless antigens like feed proteins or beneficial bacteria, so the immune system isn't constantly overreactive. When it's out of balance, this is when we get problems. So when the gut becomes irritated or imbalanced, it goes into overdrive. So dysbiosis, feed irritation, coccycycline can all trigger a continuous low-level inflammation. And when that happens, the immune system begins consuming large amounts of energy and nutrients that would normally be used for growth or egg production. This is why birds with a chronic gut inflammation often show reduced feed conversion, uneven weight, slower recovery after stress, and in layers, sometimes a reduced persistency of lay, particularly when it comes to our shell quality. It's not that the feed is poor, it's the fact that the bird is redirecting those nutrients to fight inflammation instead of building tissue and muscle. And over time, the chronic stimulation can weaken an immune tolerance. We know it when we get, when we're under chronic stress, we get run down, we get tired, we get sick. No different when it comes to the intestinal tract. This means the birds become much more reactive to minor stresses and much more vulnerable to secondary infections. So this slide shows essentially what happens when the gut integrity fails, or a condition what we call leaky gut. Under normal circumstances, that intestinal lining that we talked about forms that tight barrier, the cells are held together, and this stops anything from passing through into the bloodstream. But when those tight junctions are impacted, and inflammation damages that mucosal lever, that barrier starts to leak, and we get this pathogenic bacteria like E. coli from the water that they've consumed, or toxins progressing into the lymphatic and into the circulatory system. And that's what we call translocation, where it moves from the gut into the bloodstream, into other organs like the liver or the heart. And then once this happens, the bird mounts a systemic response within their immune system. So what began as just an intestinal issue has now become a whole bird issue. So clinically, we see this as like septicemia, yolk peritonitis, respiratory flare-ups, often following an enteric stress event like a feed change or a high protein diet, or, you know, we're seeing it a lot in more free range operators because the birds are outside and they've got access to, you know, whatever's outside. And so this is probably the most critical part of this, is managing gut inflammation so it doesn't become a septic infection, and we don't end up with these mortality events. So that brings us back to sort of the big picture. So the gut, it isn't just a tube for processing feed. It's the bird's largest immune system. It's the main site of nutrient absorption. It's a main line of defense against disease. When the gut's healthy, we get good immunity, stronger performance, less of those sort of secondary infections starting to come. Every feed decision, so ingredients, hygiene, texture, nutrient balance, has a direct effect on the microbiota, the gut pH, and the mucosal health. So making sure we've got the right mix of starch, protein, and fiber shapes that microbial fermentation pattern and helps maintain that gut barrier. Feed hygiene is really critical. Mycotoxins, rancid fats, microbial contamination sometimes can be quite overlooked as causes of gut instability. But even low-level mycotoxin exposure can do some damage to that villi and suppress that local immunity. And then the other thing is our feed form and our particle size. And I think I have a slide coming up in and how it influences our gizzard function. So remember that gizzard acts as that engine room and the powerhouse. So coarser feed stimulates that grinding and the acidification, versus a more finer feed results in that gizzard not getting the acidification right, meaning that we don't get the breakdown of this feed into those easily absorbed characteristics. So using feed additives strategically, not reactively, is probably the most, you know, key part. We want to start with like probiotics, so live microbes that colonize the gut. One of the biggest parts of probiotics is starting it from day one. Remember that early colonization of bacteria. It is critical that we're putting microbes in when the gut has the ability to colonize with those microbes. Otherwise, they're going to be competing for the already colonized bacteria. So using the right probiotic strains, producing their short-chain fatty acids, lower gut pH, even potentially modulating our immune response through our gut system, we've already sort of discussed. But our prebiotics, these are our non-digested fibers. Think of them as microbe food. So they feed that beneficial bacteria within the gut. They enhance that growth of the lactobacillus that is, you know, critical for acidifying the gut, and improves that gut barrier function. Now, how do you know which one to use? And there's like, you know, there's thousands on the market. The one thing I can suggest is get the data, read the data, and sense check the data. If they don't have peer-reviewed information, or information where, you know, there's good p-values or good statistical analysis, then you need to ask more questions. And don't be afraid to ask the questions and dig deeper until you get the answer. I've already been through that. So the particle size and the feed structure, what was really interesting at a conference not long ago, they talked about giving the largest sized feed particle to the bird that the bird can handle. And this goes from day one. Coarse particles, generally above that 1 millimeter, stimulates that gizzard development and prolongs that digestion retention time, allowing that digestion to be absorbed over a longer period of time. So when the gizzard works properly, those nutrients are exposed to the digestive enzymes for longer, improving our digestibility of protein, and then stopping that excess protein reaching our seeker and creating a dysbiosis within our microbiota there. Where, on the other hand, diets that are too fine or too powdery, often from either overgrinding or excessive pelleting, lead to really weak, poor gizzards, faster digestion passage, higher gut pH, and then we end up with things down in the seeker that we don't want. So recognizing and managing gut imbalance. Now, this often happens quietly. We don't tend to see it. It usually happens following a diet change or coccidial cycling or excess protein load, or you've just let the birds out of the shed if you're free ranging. And you'll notice subtle things first. You'll notice like wetter droppings, you'll notice uneven weights, birds just slightly off their feed, maybe a drop in water consumption. Prevention shouldn't be complex though. It's about just consistency. So keep the diet steady. If you're introducing one type of protein or one type of fat in the first ration, make sure you're feeding it through the rest of the rations, even if it is at a low diet. One of the worst things that you could do is add a fat in at the third ration that they haven't had exposure to in the 2 rations prior. Now, the reason for that is the microbiota that they are creating, they're constantly redeveloping this microbiota, and they're developing it based on the feed that they have exposure to. So if they don't have exposure to a particular raw ingredient in the starter ration, and then in the developer ration, they all of a sudden get introduced to it, they don't have the microbiomes to break down that particular raw ingredient. And so that's when you can get a little bit of inflammation and a bit of dysbiosis coming to the forefront. Now I've got some real world scenarios, one broiler, one layer. We've got on a broiler site, we had a shift to a finer pellet, and we removed the whole wheat from the diet. It improved the uniformity of the feed through the mill and it increased throughput through the mill, but it reduced the gizzard activity on farm. And what I mean by that is the birds showed a much faster digestive passage, and we ended up with feces looking like this. We ended up with wetter litter, as you can imagine. This was full of water, and we ended up with poor gizzard and proventricular health. So this is our proventricular here and our gizzard, and it was about the size of my palm. So probably 2 or 3 times the size of what it should be. What happened here? Well, by removing our whole wheat and moving to that finer pellet, we completely changed how that gut worked, not just what it digested. Without those coarse particles and without that whole wheat, the gizzard completely lost its ability to be stimulated. It wasn't contracting, which meant the muscle was deteriorating. And feed then literally just became, this just became one big tube. So it went from the crop through the proventriculus, through the gizzard and into the intestines without really anything happening within that, within this powerhouse. So the pH in the gizzard rose because, again, there was none of that enzymatic activity occurring and none of that mechanical breakdown. It didn't reduce the pathogen control because, again, our pH started to climb, which meant that more undigested material reached the lower gut, fed the wrong bacteria, compromised the mucosal barrier, and within days we started seeing some rises in mortality. What did we do? Well, we reintroduced the 10 to 15% whole wheat almost immediately to try and get that gizzard functioning properly. We adjusted our pelleting to have a little bit, a much larger pellet, not make it as sort of powder fine. We did a rejig of our dietary protein to see whether we were getting too much protein being passed through to the seeker. We added a 3-day organic acid program just to sort of give that gizzard a bit of a boost to drop that pH down and hopefully get some more digestion happening. Our litter dried up within a couple of days. Our gut tone restored within about a week or 2. But again, what happens if this damage wasn't necessarily nutritional, but it was biological? Now, this goes for both our layers and our breeders. All our chickens get coxy. Even in our vaccinated flocks, cycling illnesses can damage that mucosal layer and trigger that inflammation. So shifting gears from our nutrition-driven gut disturbance to our pathogen-driven, basically coxy is a protozoan parasite, invades those mucosal cells of that intestine. It then results in lots of multiplication. This is the life cycle here. Lots of multiplication where it goes into the enterocytes, multiplies, exit out. As it exits out, it takes a piece of the intestine. It's like breaking open a wall. It takes a bit of that intestine with it, goes into the next enterocyte, replicates, and on it goes again. Takes about 7 days, but these eventually get passed out the manure. Bit of warmth, bit of moisture, allows these oocysts to become infective. The next bird comes along and eats it up. So within, a couple of rounds, you can have a tenfold increase in the amount of coccidiosis available in the environment. Now, that's not necessarily a problem if it's managed well. It is a problem if that cycling occurs either too rapidly, meaning that you get too much of an increase of oocysts becoming infective and overloads the chicken's ability to mount an immune response to, or if you don't get enough of cycling. Again, if you don't get enough cycling, you don't get an immune response at all, and the bird doesn't develop that innate immunity to it. Some of the examples of cocci that we've seen is our E. cervellina creates these white striped lines on the serosal surface of the intestine. Our E. maxima pinpoint little dots along the intestine, creates a soft or orange mucus within the droppings. This one has a particular severe impact on nutrient absorption. E. tenella, only occurring within the ceca, creates massive hemorrhage. Don't worry about what it does to your performance, because high mortality is probably the most critical part of it. And then for our breeders and our layers, our necatrix, upper and lower mid-intestine, covered with these little white pinpricks or plaques, often referred to as salt and pepper. They're essentially hemorrhages, and so we end up with this horrific enteritis and a really high mortality, as you can imagine. Now, after a cocci challenge, even a mild one, we often see a secondary bacterial issue. So that's E. coli, Enterococcus, Clostridium perfringens, because they all thrive in this damaged environment. Now, keeping in mind that the same process of gut health and overall health plays out not only in broilers, but also in our long-lived birds, such as our layers and breeders. So repeated minor insults, whether that's the coccicycline, dysbiosis, heat stress, slowly and gradually weakens that mucosal barrier. Over time, that leads to chronic inflammation and what we call that leaky gut. So E. coli or Enterococcus cross that barrier, we start to see completely different symptoms. Not necessarily gut lesions, but peritonitis, thin shells, production losses. So another example, real world example, flock of 20,000 Isaac Browns, 45 weeks, started noticing increased seconds and thin shells and rising peritonitis post-peak of production. Our feed and water take were completely normal. What was happening inside the birds? Well, we started to see some mild enteritis signs. So a little bit of inflammation, little bit of sort of fragility within the intestine. It wasn't holding form. We were obviously seeing some reduced calcium absorption because the shell strength was really starting to deteriorate despite them only being 45 weeks of age. And we started seeing a lot more peritonitis, which is, our worst nightmare when it comes to layers. Leaky gut allowed E. coli essentially to translocate and translocate into the abdomen, into the liver, and into the heart. So how we fixed it? We replaced some of that fine limestone with a much coarser or 2 to 4 millimeter limestone to try and give the gizzards something to grind, because layers typically run a mash or a crumb feed, and it just wasn't enough for this gizzard to be doing anything. So adding a bit of coarse source of limestone did two things. One, gave the gizzard something to do, but two, also stayed longer in the gizzard and provided a much steadier release of calcium overnight. Rather than the coarse limestone that provides this real peak in calcium serologically. We also added a small wheat fraction to try and bring back some of that gizzard activity. And then we used a short program of probiotics and organic acids to help repopulate some beneficial bacteria. And the acids lowered that pH to really support that probiotic supply. We're trying to reduce that E. coli pressure essentially. So when gut barrier fails, the hidden pathway is to peritonitis or to some septicemia. Enterococcus in broilers is another classic example of where the gut barrier causes some more systemic issues. So essentially our take-home messages: the central role of the gut is ultimately in the immune system. It's in the structure, it's in the microbiota that dictates the overall health and performance of our birds. We need healthy villi, we need balanced microbiota to enable that efficient absorption of proteins to maximize our productivity in our layers, in our broilers. Essentially, finally, really, we've got an integrated management. So there's no single solution to any of this, but it's a combination of diet, environment, additives, hygiene, monitoring that protects the gut. So whether you're managing broilers, layers, breeders, any of our feathered friends, protecting the gut, specifically the immune system of that gut, is going to be where you get the most benefit out of this. And I think that's me done.